Okay, so welcome everyone to the 58th lecture of Dr. Hydri Step 1. Before we begin our lecture, can we make sure that you guys can hear my voice? If you guys can hear my voice, can I get a yes, please, on the chat box? Okay, good, very good. Okay, first and foremost, let me wish you guys a very happy new year and hope you guys have a very good New Year's Eve today. The next time I will be seeing you guys is exactly next year. That is on the 1st of Jan tomorrow. And hoping that from next year onwards, we can begin this year fresh with the optimism and goal that each and every one of us who are willing to sit for the exam, that is USMLE Step 1, we do extremely well and we get the scores that we are earning for and hopefully land that residency, which we are hoping to land. With that being said, first of all, let me apologize for starting the lecture a bit late at 9:30. We had to be honest, there were some personal problems that I had to deal with and come over here. So my apologies. But hopefully from tomorrow, the lectures again will be conducted at from 9 a.m. onwards. With that being said, did you guys get some chance to do the URL questions yesterday from biochemistry? That is from the topics which we discussed yesterday. Okay. How many questions did you guys do? How many questions did you guys do? 80 questions. Okay. 80 by Dr. Hosam. Who else? How many questions did you guys do yesterday? "40 questions and 6 wrong." That is 34 out of 40. That is extremely good. That is a very, very good score. Did you do these questions ever before, or was this the first time that you did these questions, Dr. Bashir? Out of the first time that you did 40 questions, you got 34 right and 6 wrong? You got 34 questions right and 6 questions wrong. Is that the statistics? Is that your stats? Was it on tutor or time mode? Okay. Well, if you did 40 questions and you got 34 right and 6 wrong, then it has been, I mean, let me tell you that it's a very, very good score. And if you can keep up with these types of scores, then you can easily score any mark above 240 in the real exam. So congratulations and very good. Really proud of you guys all. Anyone else who wants to give me any other good news? Oh, this is your second time reviewing first day. Very good. Okay. Is there anyone else who wants to give me any other good news regarding their questions? How many questions did you guys do? And how many questions did you guys get right? Okay. No problem. So let's not waste any time. And should we begin with our lecture today, or should we do the revision and recapitulation first? Really quickly, let's do the revision and recapitulation from yesterday's lecture. Are you guys ready? Are you guys ready with the revision and recapitulation? Okay. So let's begin. First of all, can you guys tell me the 4 adverse effects of ethanol metabolism? The 4 adverse effects of ethanol metabolism. Ketosis, very good. lactic acidosis, hypoglycemia, and hepatosteatosis. Very good. Okay. Okay, good. Very good. If you want to prevent alcohol overdose, which medication will you prescribe to your patients? Okay. If you want to discourage someone from drinking excessive alcohol, which medication will you prescribe? Okay, good. Okay. Next one is um, you have a patient who has impaired wound healing, bleeding from the gums, and which step of collagen synthesis will be affected? Step 2, that is hydroxylation. Okay. If you have vitamin C toxicity, what will be your state of iron in the body, serum iron? Okay, good, very good. Okay. Can you guys tell me the names of the soluble vitamins? What are the names of the fat soluble vitamins? Vitamin A, D, E, K. Very good. Okay. What are the toxicities of vitamin A? Vitamin A toxicities. Okay, good. Very good. Did you guys get a chance to go through the pictures with Dr. Jordan, whom I want to thank once again, which Dr. Jordan posted in the Facebook discussion group? Okay. Okay, good. Very good. All right. What are the deficiencies of vitamin A? Okay, good. What are the 4 enzymatic reactions or the 4 enzymes where vitamin B1 is used as a cofactor? Pyruvate dehydrogenase, alpha keto acid dehydrogenase, branched chain amino acid dehydrogenase, and transketolase. Very good. Okay. Okay. What are the reactions where vitamin B3 is used? Okay, good. What are the reactions where vitamin B6 is used? Vitamin B6. Okay, good. Okay. Which 2 vitamins do you need for the synthesis of niacin? Okay, very good. Then the next one is, what type of disease or problem will arise in vitamin E deficiency? Vitamin E deficiency. Okay. What type of problem would arise in a patient with vitamin E? Okay, good. Very good. What are the clotting factors that are dependent on vitamin K? Okay. If you have megaloblastic anemia due to folate deficiency, what are the lab findings? Okay. What are the lab findings of the patient with megaloblastic anemia due to vitamin B12 deficiency? Okay, good. In both B12 and B9 deficiency, what would happen to the serum cysteine level? Increased. Very good. Okay. What would happen to the serum cysteine level if vitamin B6 is not there? Okay. Which nucleoside is synthesized from homocysteine and methionine and that reaction from adenosylmethionine? Which nucleoside is synthesized from homocysteine? Adenosine. Very good. Adenosine. Okay. If you have a patient with megaloblastic anemia after in 3 to 4 months, what type of deficiency is this? B9 or B12? B9. Okay. B9 deficiency. Okay, good, very good. So from what it seems, you guys are doing a perfectly good job of recalling yesterday's lectures. And how do we feel in terms of the vitamins? Do we feel confident, yes or no? Okay. What is the best way to revise the vitamins? What is the best way to revise the vitamins? Questions. Questions, and to be honest, the picture mnemonics from the physio will also really help you guys. So if you guys forget which picture depicts which information, all you have to do is review the lecture once again, which we have given it to you. And keep in mind the lecture which we have provided for you, you guys can have a lifetime access of that lecture from our Google Drive anytime you want. Even let's say in the future, you want to use the lecture, you can use it. Or if you need it for any other exam, then you can use it too. Next question, Doctor, was yesterday's lecture deleted from the link? Yes, yesterday's lecture was deleted from the link and was uploaded on the Google Drive. It's uploaded on the Google Drive in the biochemistry Google Drive, which we have provided for you. So if you can go to the Google Drive over there, you can see that we have uploaded the lectures over there so that you can access it for your lifetime. Okay, lifetime access. None of the lectures will be removed ever. Everything will be there in the Google Drive forever. Okay. Everything will be there in the Google Drive forever. Okay. Good. So with that being said, can we begin with today's topic? That is biochemistry metabolism. Okay, good. Let me share my screen. Okay. Okay, first and foremost, can you guys see my screen over here? Can you guys see the screen? Can you guys see my screen? Okay. Are we ready to begin with biochemistry metabolism. Okay, let's begin. So today we're going to start with a very difficult topic, but to be honest, if we understand how the pathways are going step by step, then it's not exactly very difficult. There are a few sets of questions which keeps on coming years and years over and over again. And if you can identify these questions, then you can see that biochemistry is actually very easy, and it's extremely easy in biochemistry if you have all the pathways in your mind, and it will actually allow you to increase your score in step one drastically, very drastically, because if you can master biochemistry, especially the metabolism pathway, then you will be above the 75th percentile of students in the world who is planning to sit for the exam. Okay. How many of us over here find biochemistry difficult? First of all, let's start with that. Okay. Biochemistry, basically, it's not very difficult. What's happening over here is that you have to understand that the food and the things which you eat, there's a certain way they get broken down in the body. The food and everything which we eat on a daily basis, they are broken down in a certain way in the body. And if all the enzymes in those pathways are working in a normal manner, then none of the diseases which are related to biochemistry would arise. So all the diseases that arises from the biochemistry, especially the biochemical diseases, right? They are all related to more or less enzyme deficiencies, meaning if there is a deficiency of one enzyme, then this will give rise to a single disease. So that's what we are going to focus on more or less. But before we do this, we have to do a short introductions before we jump into the pathways over here. Now, let's talk a little bit about the pathways. The pathways, they can be very overwhelming. Okay, they can be very, very overwhelming. But after we are done discussing the pathways today, what I need you guys to do is that this page of first aid, okay, this page of first aid, I need you guys to print this page out. Okay, hear me out. I need you guys to print this page, and in your study room or in your library or wherever you study, I need you to have this page in front of you. Do you understand what I mean? I need you to have this page in front of you so that whenever you sit down in front of your desk, you see this page and you do a quick 1 to 10-second revision, just looking through the page. You just look through it. So what happens is when you look through the page throughout the whole month and month after month, you do the same thing whenever you start studying. Next time when you get a question, you can be very confident that which step they're talking about. To be honest, you do not need to memorize each and every step, exactly what's going on over here, because questions are very particular to a certain step, right? So you have to understand and target which step they're talking about. They might target this step. They might target this step and ask you a question, or they might target this step over here and ask you a question. So it's extremely important for you to print this page out, print out of this page and post it on your wall in your library or in your study room. So that's that. Okay. So without further ado, let's begin. The first thing that we're going to talk about today is biochemistry metabolism. And before we begin, we have to understand that there are some metabolic sites. Okay, let's talk about this in a blank page before we come over here. Metabolic sites, meaning that in the cell you have mitochondria and you have cytoplasm. There are different types of reactions which goes on in the mitochondria, and then there are different types of reactions which goes on in the cytoplasm. And this is a very favorite question in the USMLE step one exam, that they want to see whether you can differentiate which reaction and which enzymes are present in which organelle. So first of all, let's talk about mitochondria. Okay, let's talk about mitochondria. What are the reactions that are occurring over here in the mitochondria? First of all, when we talk about mitochondria, do you guys remember that when we studied science in general, we realized that mitochondria was the powerhouse of the cell? Yes or no? Powerhouse of the cell, right? Okay, if mitochondria is the powerhouse of the cell, then can we assume that all types of heavy reactions, heavy reactions, for example, breaking down complex stuff and everything else, they should happen in mitochondria? Yes or no? Okay. Okay. Now, if I talk about burning fat and burning carbohydrate, which one is more complex? Fat breakdown or carbohydrate breakdown? Burning fat, right? So mitochondria, the first one that mitochondria is involved with is fatty acid oxidization. Fatty acid oxidization. Next time, if you come across a question where they ask you fatty acid oxidization, which organelle does which organelle conducts fatty acid oxidization? Can it be easy for you to remember that we had this discussion today that mitochondria and cytoplasm, when we compared this, we said that mitochondria is the powerhouse of the cell. And since it's the powerhouse of the cell, it will be involved in the breakdown of complex substances, for example, fatty acid. So do you think there will be a confusion in the future when you think about fatty acid oxidizations and its organelle? No. Okay, so this is the thought process which I want to invoke in you all, that this is the thought process which you should invoke in yourself instead of blindly memorizing what's happening over here. Next one is TCA cycle, that is ATP production. We also know that mitochondria is involved with ATP production. Am I correct that it provides ATP? Yes or no? Yes. So TCA cycle will 100% occur in mitochondria. Then another source of energy is ketone bodies. That is, do you realize that in anaerobic metabolisms, we have ketone body formations, right? And the brain uses those ketone bodies in terms of starvations and everything else. Yes or no? Can we get some fast answers? I will need some fast answers from you all today. Okay. So if that is also a source of energy production, can we also assume that mitochondria is involved with ketogenesis? Ketogenesis? Yes? Okay. Next one is oxidative phosphorylation. Oxidative phosphorylation. Oxidative phosphorylation is another type of enzymatic reaction where via the process of oxidative phosphorylation, you have the formations of the ATP. That is, ADP is phosphorylated to form ATP. So can we assume that oxidative phosphorylation is also occurring in mitochondria? Yes? Okay. Oxidative phosphorylation. Next one is after we break down glucose or any other substance for that matters, do you realize that when you break down glucose, when you break down carbohydrates, proteins, and fat, you have a production of a substrate which is responsible for entering the TCA cycle and is responsible for the formation of ATP? Yes or no? Everything gets broken down to this substance, and then they enter the mitochondria for ATP production. So can we assume that that substance which we are talking about is, does anyone have any idea which substance are we talking about over here? Anyone? Fast answers, please. Acetyl-CoA, very good. Dr. Midula, very good. Acetyl-CoA. Acetyl-CoA, okay. Now, this is the most logic way to understand the functions which are taking place in the mitochondria. If we are faced with any questions in the future about these pathways, whether they're happening in mitochondria or whether they're happening in cytoplasm, can we be 100% confident that it's happening in mitochondria? Yes or no? Okay. Okay. Good. Very good. Okay. Okay. Next one. Next one is cytoplasm. Cytoplasm. Before we talk about cytoplasm, can we talk about the reactions which are happening in both mitochondria and cytoplasm? Both. Well, what are the reactions which are happening in both mitochondria and cytoplasm? You know what mitochondria does to the cytoplasm is mitochondria. Does anyone know what mitochondria does to the cytoplasm? Let me see if I have invoked any type of what can I say? any type of structural intelligence in you guys. Mitochondria. Does anyone know what mitochondria does? Mitochondria dash cytoplasm. What does mitochondria do to the cytoplasm? Mitochondria hugs. Nope. Mitochondria hugs cytoplasm. Okay. Mitochondria loves hugging the cytoplasm. This is a mnemonic. Okay. This is a mnemonic. Mitochondria hugs the cytoplasm. So if we talk about mitochondria hugging the cytoplasm, then the reactions which are happening in both mitochondria and cytoplasm is, first of all, heme synthesis, then urea cycle, then gluconeogenesis. My apologies. Gluconeogenesis. Okay. S is silent over here. There's no reaction with S, so that's that. So heme synthesis, urea cycle, and gluconeogenesis. So these are the three reactions which are occurring in both mitochondria and cytoplasm. Are we all clear about this? Yes or no? Yes or no? Now, can we assume that all the other reactions in the body, except these 5 and these 3, that are solely occurring by themselves, they're occurring in the cytoplasm? Yes or no? Cytoplasm. Cytoplasm is all the other reaction. All other reactions. Okay. How easy has this been? Out of 1 out of 10, how easy has this been to understand? Okay, very easy, right? Okay, so how do we remember this instead of blindly memorizing? First of all, when we think about mitochondria, think about mitochondria being the powerhouse of the cell. Since they're the powerhouse of the cell, they can take the load of metabolizing the complex stuff. So the most complex stuff to metabolize is fatty acid. And they're also responsible for production of energy. So they have to conduct the TCA cycle where they produce the energy. And in the absence of proper aerobic metabolism, they're also responsible for forming the ketone body. So ketogenesis, oxidative phosphorylation is the production of ATP from ADP and another substrate that will be produced over here. So the acetyl-CoA production over here, meaning that everything that is broken down in the body leads to the formation of a substrate acetyl-CoA, which will enter the TCA cycle to produce ATP. So acetyl-CoA is also produced over here. Next one we talked about was mitochondria hugs the cytoplasm, that is heme, urea, and gluconeogenesis occurring in both mitochondria and cytoplasm. And all the other reactions in the body that are solely occurring by themselves occur in the cytoplasm only. So if we talk about glycolysis, where does glycolysis take place? Glycolysis. Cytoplasm. HMP shunt. Cytoplasm. Synthesis of cholesterol, proteins, fats, and nucleotides. Every synthetic pathway. Synthesis of all substances occur in the cytoplasm. Okay. Okay.
Someone is waiting.
Someone is waiting. Okay. Okay. All right. Okay. Okay. So with that being said, have you guys heard of the HMP shunt pathway? HMP shunt pathway. Yes. HMP shunt pathway. What is it responsible for? What does HMP shunt pathway? What does it produce? That is responsible. Is that a source of energy too? Is that also a source of energy, which HMP shunt pathway, which it produces? Okay. HMP shunt. Okay. So what happens is, do you know what cytoplasm tells to mitochondria? Do you know what cytoplasm says to mitochondria? When cytoplasm sees that mitochondria is working so well in producing all the energies, right? They're conducting the TCA cycle, they're conducting ketogenesis, they're conducting oxidative phosphorylation and acetyl-CoA production. Cytoplasm feels bad for mitochondria, and cytoplasm goes and tells mitochondria that, okay, instead of you taking the load of making all the different types of energy, let me conduct one pathway, which will allow me to produce some energy too. Mitochondria asks, which pathway will you conduct? And cytoplasm answers that the pathway I will conduct is HMP shunt pathway. So next time when you are thinking about energy pathways, think about the fact that cytoplasm feels bad for mitochondria. So cytoplasm will conduct the HMP shunt pathway, and mitochondria will not conduct the HMP shunt pathway. Mitochondria will conduct all the other pathways except HMP shunt when it comes to energy production. Are we clear about this? Yes or no? Okay. Okay. So has this been easy, or if I asked you guys to blindly memorize all the pathways of mitochondria and cytoplasm, would that have been easy? Okay. Easy or not? Do you guys understand? Okay, so with that being said, let's go back to our text. Let's see what's going on over here. So mitochondria, first of all, this is very high yield. Okay. Mitochondria is fatty acid oxidation, acetyl-CoA production, TCA cycle, oxidative phosphorylation, and ketogenesis. We talked about these things. Cytoplasm will conduct glycolysis, HMP shunt, and synthesis of cholesterol, proteins, fatty acids, and nucleotides. Always remember that cholesterol will be produced in the smooth endoplasmic reticulum. This is high yield, cholesterol in the smooth endoplasmic reticulum. Proteins will be produced in the rough endoplasmic reticulum, and we talked about this, fatty acids and nucleotides. And both organelles, mitochondria and cytoplasm, will conduct HMP, heme synthesis, urea cycle, and gluconeogenesis, because mitochondria hugs the cytoplasm. Okay, are we clear about this? Yes or no? Okay, good. Okay, so let's move on to the next page. Okay, let's begin with this one. Okay, with this portion of the table, this one, this is the portion of the table which we will discuss later. I'm going to tell you why, because in order for us to understand these enzymes and these processes, we have to understand the pathways first. So we will learn the pathways first, and then we will come back to this table, and we will learn about this table. How does that sound? Does that sound okay? Yes or no? Okay, because we have to learn the pathways first, and then we will come back over here. So let's skip this table for now, and let's talk about enzyme terminology. What do we mean by enzyme? Different types of enzymes by the names which they have. Enzymes terminology means that an enzyme's name often describes its function. For example, glucokinase is an enzyme that catalyzes the phosphorylation of glucose using a molecule of ATP. Kinase basically. So let's talk about each, and I think we skipped another table too in genetics. Yes, we did. Which one did we skip? Which one did we skip? Which one did we skip? Dr. Osam, do you have any idea which one we skipped in genetics? I'm pretty sure I talked about this. Page 62, the one with summaries. Page 62. Oh, trinucleotide repeats. Yes, but we're not done with trinucleotide repeats as of yet, right? We're not done with Huntington's and Friedreich's ataxia, so that's why. Okay. Okay. Very good job. But to be honest, we're not done with Huntington's, because we have to talk about Huntington's and all the other one, right? Okay, you scared me. I thought that I completely forgot to talk about a table. All right. So right after we are done talking about Huntington's and Friedreich's ataxia, we will go back to that table. Okay. So can we begin with enzyme terminology? All right. As a matter of fact, what we would do is after the end of today's lecture, we will talk about Huntington's and Friedreich's ataxia and get done with that table. How does that sound? Okay. That's what we're going to do. It will be very easy for us. Okay. Enzyme terminology. The enzymes, the following are commonly used to use enzymes as their descriptions. First of all, is kinase. What do we mean by kinase? Kinase is basically the transfer of a phosphate group from a high energy molecule to a substrate. This is very easy to understand that kinases are basically the enzyme which will transfer a phosphate from a high energy phosphate group to another substrate. That's kinase. Kinase is the transfer of a phosphate group. Very easy to understand. Next one is phosphorylase. Phosphorylase is addition of inorganic phosphates onto substrate without using ATP. So one is the transfer of a phosphate group. Another one is addition of an inorganic phosphate onto substrate without using ATP. So you do not use ATP when you use phosphorylase. Phosphorylase is the enzyme which does not need the use of ATP. The enzyme phosphatase means that there is removal of the phosphate group. That's very easy to understand. Whenever an enzyme has ASE in the end, it removes the phosphate group. Next one is dehydrogenase. Dehydrogenase, the word itself means that there is a catalyzation of oxidation and reduction reactions happening at the same time. So oxidation and reduction. So let's focus on this four. First of all, kinase is addition of a phosphate group from a high energy molecule. Phosphorylase is addition of inorganic phosphate. Phosphatase is removal of the phosphate, and dehydrogenase is catalyzation of oxidation and reduction occurring together. Next one is hydroxylase. Hydroxylase is addition of hydroxyl groups onto substrates. Hydroxylation is addition of hydroxyl groups. Carboxylase is transfer of carbon dioxide groups with the help of biotin. Do you guys remember yesterday when we talked about biotin B7, we talked about the fact how it's used for the 3 types of carboxylation reactions, yes or no? Can you guys tell me the 3 carboxylation reactions where biotin is used? Okay, good. Very good. Next one is mutase. What is mutase? Mutase is relocation. It relocates a functional group within a molecule. It relocates a functional group within a molecule. For example, vitamin B12 dependent methylmalonyl-CoA mutase. It relocates, it takes a functional group from one position and it relocates that functional group to another position. So that's mutase. Synthase and synthetase are basically joining of 2 molecules together with the use of energy. Okay, how will you receive these questions over here? First of all, the questions, these questions, they are not extremely high yield. They are a relatively of a lower yield question. The places where you will receive these questions are different types of NBME. These are NBME questions. NBME questions, the questions will come from this side of the table, and the answers will be the names of this one. So basically, if you have to test yourself, you can cover this part with your hand and read this part and see whether you can understand. Okay. So can I give you guys 2 minutes to read this once again? And I know if I ask you guys these questions, it will be extremely easy for you to answer. So 2 minutes just to read this for once, and then we can move on. Yes or no? Can I give you guys 2 minutes to read this? The different functions of different enzymes. So it's 10:11 as of right now. I'll give you guys 2 minutes at 10:13, and I'll ask you guys the questions. Can I get a yes or no, please, to see if we're all on the same page? Okay, good. So I'll give you guys 2 minutes. It's 10:11, and let's start with the questions at 10:13. Okay. Okay. So what is the name of an enzyme which is responsible for catalyzing the transfer of a phosphate group from a high energy molecule? Fast answers, please. Kinase. Name of an enzyme which is responsible for addition of inorganic phosphates. Addition of inorganic phosphates. Phosphorylase. Name of an enzyme which is responsible for removal of a phosphate. Removal. Phosphatase. Catalyzation of oxidation and reduction reactions. Okay. Next one is addition of hydroxyl groups onto substrates. Transfer of carbon dioxide groups with the help of biotin. Biotin. Very good. Relocation of a functional group within a molecule. That is, they take one molecule from one location and take it to another location. They mutate it. Mutase. Very good. Name of the last enzyme which is responsible for joining the two molecules together. Synthetase or synthase. Okay. Very good. Okay. Fatty acid oxidation. Which organelle conducts fatty acid oxidation? Which organelle conducts urea cycle? Which organelle conducts gluconeogenesis? Which organelle conducts HMP shunt pathway? HMP shunt pathway. HMP shunt. Very good. Which organelle conducts gluconeogenesis? Both. Very good. Not cytoplasm. Cytoplasm will conduct glycolysis, not gluconeogenesis. Okay, so how about synthesis of cholesterol, proteins, and proteins and fatty acids? Cytoplasm. Okay. TCA cycle. TCA cycle. Oxidative phosphorylation. Acetyl-CoA production. Okay. Are we all clear about this? Yes or no? Okay, good. So let's begin with the pathways. Are we ready? Okay, let's begin with the pathways. The first pathway that I want to talk about, from where all the other pathways are usually coming, the first pathway that I want to talk about is the glycolytic pathway. That is the glycolytic pathway over here. That's what I want to talk about. Okay. So before I talk about the glycolytic pathway, let me see one thing. Okay. Let me see one thing over here. I think I have a better pathway over here. Just give me 1 second. Okay. So can you guys see my screen over here right now? Okay. So this pathway is a complete pathway. The pathway that is in the pathway that we have over here in first aid, that pathway is not a very complete pathway. So what I need you guys to do is I need you guys to I'll post this one in the group. Okay, I'll post this in the group, and then from the group we will talk about how it's better for you to print this page out. Okay, you will print this page out, and then you will keep this page in your library or in your study room, where you can learn about this. Okay, instead of using the pathway in the first stage, I need you guys to use this pathway. Is that okay? Okay. So the first pathway that we're going to talk about is the glycolytic pathway. Just give me 1 minute. Okay, the first pathway that we're going to talk about is the glycolytic pathway. Okay, so glycolytic pathway. First of all, glycolytic pathway, which organelle conducts the glycolytic pathway? Cytoplasm. Very good. Okay, so cytoplasm will conduct the glycolytic pathway. So glycolysis, the word glycolysis, what does that mean? That glycolysis basically means breaking down of glucose. Glycolysis is the breakdown of glucose, glycolysis. Okay, so step by step, that's what we're going to do. We're going to talk about the pathways. And so the first step of glycolysis is, first of all, you have glucose. Okay, you have glucose. Glucose over here, when we have carbohydrates are broken down. They're disaccharides. I mean, they're polysaccharides. After that, they are broken down to monosaccharides, which are glucose. So glucose is broken down to the first substrate by the enzyme glucokinase, or hexokinase. When we talk about glucokinase or hexokinase, what molecule are they adding to the group, to glucose? Addition of phosphate. So if they add phosphate, do you remember how we talked about the fact that addition of phosphate requires the transfer of the phosphate group from a high energy molecule to a low energy molecule? Yes or no? Yes. Okay. So that's that. Glucokinase or hexokinase. So what they will do is they will add it from a high energy group to a low energy group. So basically over here you have ATP, and they are being broken down to ADP. Okay. Then if we add phosphate to the glucose, do you know at which position of the glucose the phosphate gets added to? The phosphate gets added to the 6th position of the carbon atom. So this is called glucose 6-phosphate. Glucose 6-phosphate. Okay. Glucose 6-phosphate, after it's formed, it easily converts itself spontaneously to fructose 6-phosphate. To fructose. What happened? 1 second. Okay. So once again, you had glucose, then glucose 6-phosphate. Over here, you have glucokinase and hexokinase. And you have the ATP, right? ATP to ADP. Okay, that's what we discussed till now. Then glucose 6-phosphate is spontaneously broken down to fructose 6-phosphate. Fructose 6-phosphate, what happens is fructose 6-phosphate, after it's formed, fructose 6-phosphate undergoes a phosphatase reaction. What happens is there is another type of enzyme. My apologies. It's not a phosphatase reaction. It's a kinase reaction. There's another addition of the phosphate group. There's another addition of phosphate group. This addition of phosphate group is known as phosphofructokinase 1. Phosphofructokinase 1. If we talk about phosphofructokinase 1, then can we assume that over here you have another addition of the phosphate group? Yes or no? So over here, will ATP be broken down to ADP again? because kinase is the transfer of a phosphate from a high energy to a low energy, right? So ATP will be broken down to ADP. And fructose 6-phosphate now has another phosphate group. So if we want to add the phosphate group right now, do you have any idea where, in which position of the carbon atom is the phosphate group being added? Very good, 1. So this is called fructose 1,6. And how many phosphate do we have? 2 or 1? 2. So it's called bisphosphatase. Bisphosphate. Fructose 1,6 bisphosphate. Okay. Fructose 1,6 bisphosphate undergoes the mechanism of breakdown into two high yield things. Into two things. Fructose 1,6 bisphosphate can be broken down, or it converts itself to two things. Okay, if you can understand up to this right now, everything will be very easy for you. Fructose 1,6 bisphosphate will be broken down to form glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. And dihydroxy acetone phosphate. Dihydroxy acetone phosphate. Are we clear about this? Yes or no? Till now? Okay. Now what would happen is glyceraldehyde 3-phosphate, it undergoes the mechanism of another breakdown, right? Glyceraldehyde 3-phosphate over here, what happens is glyceraldehyde 3-phosphate results in the formation of 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. What happens is over here you have, okay, so let me just repeat the whole thing from again. Glucose is broken down, glucose is formed into glucose 6-phosphate by the addition of glucokinase, so ATP is used. Glucokinase converts glucose to glucose 6-phosphate, glucose 6-phosphate from fructose 6-phosphate. From here, you have another kinase adding the enzyme, adding the phosphate, turning fructose 6-phosphate to fructose 1,6 bisphosphate. Now, fructose 1,6 bisphosphate converts itself to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Are we clear about this? Yes or no? Okay, yes. Okay, now, what happens is 1,3-bisphosphoglycerate, this breaks down to form. Now the rest of the steps are very easy and not very high yield. What happens is you have 1,3-bisphosphoglycerate, which gets broken down to 3-phosphoglycerate. 3-phosphoglycerate. So over here, if they are losing one phosphate, can we assume that over here you have the formation of an ATP? Yes or no? You have the formation of an ATP, yes. So they're losing one phosphate, so over here ATP is produced. Okay. Then after that, 3-phosphoglycerate gets broken down to 2-phosphoglycerate. 2-phosphoglycerate. The amount of phosphate over here is still the same, but the location of the phosphate has changed. And instead of the third position, it's now the second position. And then what happens is 2-phosphoglycerate converts itself to phosphoenol pyruvate. Phosphoenol pyruvate. Now, if phosphoenol pyruvate, if we have the formation up to here, then what happens is the next step is very easy. Phosphoenol pyruvate is converted to pyruvate. Pyruvate, the last substrate before it enters the TCA cycle. Pyruvate. The enzyme that is responsible for this one is pyruvate kinase. Now, if there's another pyruvate kinase, can we assume that over here ATP is used up? Right? Okay. But what happens is over here, instead of adding, instead of forming an ADP, what happens is in this step, energy is produced. This is the part of the reaction where it gets a little bit confusing. Over here, you have ATP production instead of ATP breakdown. You have ATP production because the pyruvate kinase over here, it uses a cofactor. It does not use the energy. Do you guys remember the cofactor which pyruvate kinase uses? Which cofactor does pyruvate kinase use? Cofactor. Cofactor which pyruvate kinase uses. Anyone? B1. Very good. B1. Pyruvate kinase uses B1. If pyruvate kinase uses B1, do you think pyruvate kinase will need will need ATP? No. So since it does not need ATP over here, ATP is spared. So ATP is actually produced over here. And after pyruvate kinase results in the formation of pyruvate, this pyruvate undergoes the mechanism of formation to acetyl-CoA. Acetyl-CoA. This acetyl-CoA that is produced from the pyruvate, this occurs by the step of pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex, it uses a lot of the cofactors, right? Pyruvate dehydrogenase complex, one of the main cofactors which pyruvate dehydrogenase complex uses is it uses vitamin B1, right? Along with this pyruvate dehydrogenase, it uses a lot of the other enzymes. Do you guys have any idea all the other vitamins which pyruvate dehydrogenase complex uses? Yes or no? Pyruvate dehydrogenase. Very good. B1, 2, 3, 4. Pyruvate dehydrogenase, it uses B1, thymine. Then it uses B2, which is riboflavin, B3, that is niacin. And then it also uses which one? It also uses which one? Which one? Guys, B5. Very good. It's not 4, it's 5. Okay, so that's that. Right after you have formation of the acetyl-CoA, what happens to the acetyl-CoA? The acetyl-CoA, it enters the TCA cycle. Okay, it enters the TCA cycle. Are we clear about this? Are we clear about this? So this is the glycolytic pathway. This is the glycolytic pathway. Okay. All right. Okay. Pyruvate. Now, let's talk about some other thing. Pyruvate will be converted to acetyl-CoA in the presence of proper Arabic substrates over here. But let's say there is an anaerobic situation going on right now. Anaerobic situation going on. Can we assume that if it's an anaerobic situation, pyruvate can also be converted to lactate? Yes or no? Yes. So pyruvate, instead of an anaerobic pathway, pyruvate can also be converted to lactate. The enzyme that is responsible for the conversion of pyruvate to lactate is lactate dehydrogenase complex. Lactate dehydrogenase. Okay. Lactate dehydrogenase. And if we talk about one enzyme that is responsible for lactate dehydrogenase, I mean one cofactor or vitamin, water-soluble vitamin, do you guys have any idea which vitamin will use LDH? We talked about the vitamin yesterday is needed for LDH. Which vitamin is needed for LDH? We talked about this yesterday. Anyone? Which vitamin is needed for the proper functioning of LDH? Dehydrogenase reaction. Dehydrogenase. B3, very good. B3, niacin. Okay, niacin. Okay, do you see how easy it is for you to correlate from yesterday's discussion which vitamin will be used for which step? Yes or no? Yes or no? Is it easy or not easy? Okay. So this is the glycolytic pathway. Do you guys have any questions for this pathway? Glycolytic pathway. Any questions? Any questions for this pathway? Can we get a yes or no, please, to see if you have understood or not understood? So that we can move on or not move on. You missed what? What did I miss? Which one did I miss, Dr. Mohammed? Dehydroaldehyde. What is dehydroaldehyde? I didn't. Oh, okay. Dihydroxyacetone phosphate. I have not talked about this as of yet. I have not talked about this pathway. Don't worry about this. I have not talked about this. I talked about the glycolytic pathway. Okay. Okay. I have not talked about this yet. I have talked about this pathway only. Okay. Next one. Cofactor. Which cofactor did I miss? Which cofactor did I miss? Pyruvate kinase. Pyruvate kinase. Over here. This one. Yes, the cofactor basically over here is, to be honest, vitamin B1 is used to some extent for the pyruvate kinase activity. Okay, but although vitamin B1 is more widely used for pyruvate dehydrogenase complex, but vitamin B1 is used to some extent for pyruvate kinase. That's why pyruvate kinase, they do not need ATP for converting phosphoenol pyruvate to pyruvate. Okay, is that clear? Have you guys understood the glycolytic pathway? All right. Okay, so if we have understood the glycolytic pathway, can we move on to the next pathway, that is the TCA cycle, to see what's happening to the acetyl-CoA in the TCA cycle? Yes? Okay. Now we will move on to the TCA cycle. TCA cycle, what happens is you have, so first of all, we have the acetyl-CoA over here. Where is this acetyl-CoA coming from? Acetyl-CoA. Where is this coming from? Very good. It's coming from pyruvate, from the pyruvate dehydrogenase complex. So you have acetyl-CoA. Now you have the TCA cycle. TCA cycle, which organelle conducts TCA cycle? So can we assume that after the breakdown of glucose in the cytoplasm, the acetyl-CoA, which is produced, it is produced in the mitochondria, and now it will conduct the TCA cycle? Yes? Yes or no? Yes. Okay. So let's talk about the TCA cycle. So what happens is the TCA cycle, okay, the TCA cycle okay, TCA cycle is basically a cycle where the enzymes, they work round and round over here, more or less. So what happens is in TCA cycle, you have you have acetyl-CoA, which comes and it enters the cycle. Acetyl-CoA, it comes and it binds with a very high yield substance. The name of this substance is oxaloacetate. Oxaloacetate. So it comes and it binds to the oxaloacetate over here. Okay. So it comes and it binds to the oxaloacetate. It binds with the oxaloacetate. So when it binds to the oxaloacetate, this one, acetyl-CoA and oxaloacetate, this results in the formation of a very high yield substrate called citrate. It's called citrate. Citrate. The enzyme that forms citrate is known as citrate synthase. Citrate synthase. Are we clear about this? Yes or no? Yes? Okay, good. So we're going to go step by step so that we do not get overwhelmed, right? First of all, once again, acetyl-CoA comes, enters the mitochondria, it's produced in the mitochondria. Then it binds with the oxaloacetate with the help of the enzyme citrate synthase to form citrate. Citrate over here, it combines with, citrate is broken down to another substance. This is known as isocitrate. So basically, citrate is isolated. So isocitrate. Do you guys know the name of the enzyme that is responsible for breaking down citrate to isocitrate? Anyone? Anyone? It's not mentioned. Very good. Aconitase or aconitase. Okay, this is not a very high yield enzyme, so don't worry about this, but aconitase is there. Okay. Okay. Cis aconitase or aconitase. Okay, let me just write it down over here. It's not a very high yield enzyme, but it's okay. Is it mentioned in first aid? Let me see. Okay, not mentioned in first aid. Okay, no problem. Aconitase. Okay. Now, next one is citrate over here, isocitrate. Isocitrate undergoes dehydrogenase reaction. And the name of the enzyme, thankfully, is the same name as the substrate. The name of the enzyme is isocitrate dehydrogenase. Isocitrate dehydrogenase. Now, if we are talking about dehydrogenase, what cofactor or which vitamin are we using over here? Which vitamin are we using? Fast answers, please. Cofactor B3. B1 should be B3, right? Niacin is used for dehydrogenase, right? Not B1. Right? B3. Because B1 is used for 4 reactions, that is pyruvate dehydrogenase, branched chain amino acid dehydrogenase, alpha keto acid dehydrogenase, and transketolase. So this one is B3. Are we clear about this? Isocitrate, after it gets after it's dehydrogenated, okay, it results in the formation of another high yield substance. It results in the formation of alpha keto glutarate. Alpha ketoglutarate. Okay. Now, this alpha ketoglutarate is converted to another substrate, meaning it's broken down over here. This is another dehydrogenase reaction over here. Most of the reactions in TCA cycle are dehydrogenase reaction. Remember that. Please remember that most of the reactions are dehydrogenase reactions over here. Okay. So the next one is alpha ketoglutarate is broken down by alpha ketoglutarate dehydrogenase. Now, if we talk about alpha ketoglutarate dehydrogenase, which enzymes are you? Which cofactors or water soluble vitamins are used? B1, very good. B1, and along with this, this is a complex. For example, pyruvate dehydrogenase was actually pyruvate dehydrogenase complex. Alpha ketoglutarate is also an alpha ketoglutarate dehydrogenase complex. Alpha ketoglutarate dehydrogenase. If it's a complex, can we assume that over here more than one water soluble vitamin is used to break down alpha ketoglutarate? Yes. So once again, the vitamins that are used is 1, 2, 3, 5. Most important one is 1, thiamine. Okay, now alpha ketoglutarate will be broken down by alpha ketoglutarate dehydrogenase to form, okay, 1 second, succinyl CoA. Succinyl CoA. Okay, they will be broken down to form succinyl CoA. Okay, now succinyl CoA is broken down. This reaction, it continues over here. It's broken down to another enzyme, not an enzyme, a substrate. This is known as succinate. Succinate. Okay, succinate. Okay. The enzyme, do you guys know the enzyme that is working over here? The name of this enzyme is, do you guys know the name of this enzyme? Yes or no? Anyone? Succinate dash kinase. What enzyme is this? The name of this enzyme is succinate thiokinase. No, it's not a very high yield enzyme. Succinate thiokinase. Okay. Succinyl CoA is converted to succinate by succinyl thiokinase. Okay, now succinate is converted to fumarate. Okay, fumarate. The enzyme that is working over here right now, this enzyme is called succinate dehydrogenase. Okay, dehydrogenase. Succinate dehydrogenase. Now, another one, again, last one is fumarate is directly converted. Fumarate is directly converted to malate. And malate is converted to oxaloacetate by the help of the enzyme malate dehydrogenase. And the cofactor is vitamin B. What? Vitamin B2, right? B2 by B3. Very good. Okay, good. So this is basically your TCA cycle. Are we clear about this? Are we clear about the TCA cycle? Okay. We're still not finished over here. Okay, we're still not finished over here because there are some things which are going on. How will be the questions? Okay, just let me finish the discussion first, then I'll discuss the questions. First of all, let me finish the TCA cycle first, okay? Okay. Do you guys remember how we talked about the fact that TCA cycle is responsible for production of the energies, right? NADs, GTPs, FADs, and then they will enter the HMP shunt, okay? So fumarate and malase, the enzyme is fumarase. It's not a very high yield enzyme. That's why I did not mention this. The enzyme is fumarase, okay? Fumarase, not a very high yield enzyme, okay? Okay. Now, do we have to or not have to know which pathways are creating the NADs, FADs, and GTPs? Yes. Is that high yield? Yes, that is extremely high yield. Okay. So the first pathway that is responsible for creating the NADs, okay, NADs are the two big dehydrogenases. All the dehydrogenases. So what are the dehydrogenases over here? For example, isocitrate dehydrogenase, right? It's converting isocitrate to alpha ketoglutarate. This pathway will create NADH. Then this pathway will create NADH. All the dehydrogenase reactions. Then malate dehydrogenase. This pathway will create an NADH. Okay. So how many total NADH is created over here? 3. Okay. Right. So 3 NADH at 3 locations. All the dehydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, malate dehydrogenase. Now, next one is FADH. FADH is produced at this dehydrogenase. That is succinate dehydrogenase. Okay. At this dehydrogenase, you have the production of FADH, not NADH. Okay, the way I would like to remember this one is F for fumarate, F for FADH. That is the best way you can remember this. Fumarate, FADH, and all the other dehydrogenases will produce NADH. So FADH. Okay. Are we clear about this? Yes or no? Okay. FADH is produced over here. And this enzyme, succinate thiokinase, I have a personal name for this enzyme. I named this enzyme as succinate. Instead of naming this thiokinase, I named this enzyme as succinate geokinase. The reason why I named this enzyme succinate geokinase is this enzyme gives us GTP. This enzyme gives us GTP. GTP. Okay, now TCA cycle is complete. Now the TCA cycle is complete. Okay, are we clear about this? Okay, this is the TCA cycle. Okay. How do you guys feel about the TCA cycle? Are you guys confident? Yes or no? Okay. Can I give you guys 1 minute to go through the TCA cycle over here? I'll give you guys 1 minute to go through the TCA cycle. And then I'll ask you questions and then we'll move on, okay? Are we clear? Then I'll ask you questions and then we will move on. Okay. 1 minute on the clock. It's 10:50. I'll ask you the questions at 10:51. Okay. Now, TCA cycle, let's talk about TCA cycle. You guys should be done with your revision right now. First of all, TCA cycle, acetyl-CoA converts to which substrate after binding with oxaloacetate? Fast answers, please. Good. Citrate is converted to which substance? Isocitrate. Isocitrate is converted to which substance? Fast answers, please. Alpha-ketoglutarate. What is the enzyme that is responsible? What is the enzyme that is responsible? Isocitrate dehydrogenase. Is NADH produced over here? Yes or no? Yes. Next one. Next one is alpha-ketoglutarate is broken down to which substance? Alpha-ketoglutarate is broken down to which substance? Succinyl-CoA. Is NADH produced in this pathway? Yes. Next one. Succinyl-CoA is converted to which substance? Succinyl-CoA. Succinate. What is the name of the enzyme? Succinate thiokinase. But what do we call this enzyme? We call this enzyme succinate geokinase. Why do we call it geokinase? Because it produces GTP. Very good. Next one. Succinate is converted to which substrate over here? Fumarate. Okay. The name of the enzyme is? What is the name of this enzyme? Succinate dehydrogenase. Very good. Is NADH produced over here? No. Very good. What is produced over here? FADH. Okay. Fumarate is broken down to malate by the enzyme fumarase, not a high yield enzyme, and then malate is converted to oxaloacetate. by malate is converted to oxaloacetate by which enzyme? Malate dehydrogenase. And then malate dehydrogenase, does it produce an NADH? Yes or no? Yes. Okay. Are we clear about this? Okay. Now, we have just got done talking about glucose. What are the two other monosaccharides that we have to talk about? Monosaccharides. We just talked about glucose. What are the two other monosaccharides that we have to talk about? Fructose and monosaccharides. Lactose. Very good. Fructose and lactose. So we will talk about lactose metabolism and fructose metabolism. Okay, are we ready to talk about lactose and fructose metabolism? My apologies. Galactose is the monosaccharide. Lactose is disaccharide. You're correct. Okay. Okay. Are we clear? Can we start with galactose and fructose metabolism? Fructose and galactose metabolism. Okay. Okay. So we're going to talk about the first metabolism that we're going to talk about is galactose metabolism. Galactose. Okay. Galactose metabolism. First and foremost, let me tell you one thing. The end product of galactose. First of all, lactose is present basically in milk. Am I correct? Yes or no? Right? Yes. So the final product of lactose breakdown is lactose is converted to galactose, and galactose is converted to a lot of things. And then finally, the final product is it's converted to glucose 6-phosphate. If it's converted to glucose 6-phosphate, can we assume that this will enter the glycolytic pathway and then get converted to pyruvate and then acetyl-CoA and then TCA cycle? Yes or no? So what is our goal? Our goal is, what is our goal? Our goal is to convert galactose to glucose 6-phosphate. This is our goal. Okay. So let's see how galactose is converted to glucose 6-phosphate. First of all, you have lactose. Lactose is, okay, so lactose, we get lactose from milk, cheese, right, and dairy products. Lactose is broken down to galactose. Galactose. The enzyme that is responsible for breaking down lactose to galactose is known as lactase. Okay. Okay. What would happen in a person who has lactase deficiency? Bloatiness, right? Diarrhea. Am I correct? Malabsorption, right? Okay. So lactase. Lactose is broken down to glucose and galactose. It's also broken down to glucose, but since we're talking about galactose metabolism, it's also broken down to glucose. And if it's broken down to glucose, then this glucose will be converted to glucose 6-phosphate with the help of which enzyme? Fast answers, please. By the help of which enzyme? Glucose will be converted to glucokinase. Very good. Glucokinase or hexokinase. Okay, now galactose over here, galactose is converted to another substance over here. This substance is known as galactose 1-phosphate. If galactose is converted to galactose 1-phosphate, can you guys name the enzyme over here? Galacto. What is the name of the enzyme? Galactokinase. Very good. Galactokinase. Okay. If galactokinase is over here, can we assume that ATP is broken down and forms ADP because kinase will transfer a phosphate group from a high energy to a low energy, right? So ATP to ADP. ADP. Okay. Now, galactose 1-phosphate that is produced over here, galactose 1-phosphate is broken down
to galactose 1-phosphate. It's broken down to glucose 1-phosphate. Glucose 1-phosphate. Glucose 1-phosphate, the enzyme that is used up over here, this is a very high yield enzyme. This enzyme is known as galactose 1-phosphate uridyl transferase. Uridyl transferase. GA1P UDT, galactose 1-phosphate uridyl transferase. This enzyme, in order to catalyze this reaction, galactose 1-phosphate to glucose 1-phosphate, what they will do is they will convert UDP, that is uridylated glucose, to uridylated galactose. They will convert uridylated glucose to uridylated galactose. They will convert this reaction. And there's another enzyme that is working over here. This enzyme is known as epimerase, or we need 4 of them. So 4 epimerase. 4 epimerase, this enzyme over here. 4 epimerase. 4 epimerase will convert UDP galactose to UDP glucose, and this reaction results in the formation of the proper functioning of galactose 1-phosphate UDP, and this is the only complex part of the galactose metabolism, is galactose 1-phosphate uridyl transferase. How many of us are over here who are learning about this enzyme for the first time in our life? For the first time over in our life, yes or no? How many of us have heard about this enzyme before? Okay. We will do a revision of this pathway. Let me finish this pathway first, because the next step of the pathway is very easy. The next step is glucose 1-phosphate is then converted to glucose 6-phosphate. Glucose 6-phosphate, the enzyme that is the enzyme that we are talking about over here, the enzyme is phosphoglucomutase. Okay. Okay, do we have any question? Are we following the Dr. Hydery Google pick, not first aid, right? Yeah, yes, we're not following the first aid Google, we're not following the first aid picture, because first aid does not have a lot of information in the pathways. The pathways that is very heavily detailed is the one from physio. Okay. Okay, good. Okay, so let me begin with the galactose metabolism again from the beginning. First one is, the goal is the conversion of galactose to glucose 6-phosphate. First of all, we get galactose from lactose. Lactose is broken down to glucose and galactose by the enzyme lactase. Galactose is converted to galactose 1-phosphate by galactokinase, which uses up 1 ATP to form ADP. Galactose 1-phosphate is converted to glucose 1-phosphate by the enzyme galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase, in order for this to work, UDP glucose has to be converted to UDP galactose, and this continues so far and so forth. And this occurs in the presence of another enzyme that is 4 epimerase. Over here, if you have a hard time remembering all this information, just try to remember galactose 1-phosphate uridyl transferase, and that will be absolutely enough. You don't have to remember anything else. After galactose 1-phosphate is converted to glucose 1-phosphate, glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglucomutase. Phosphoglucomutase. Mutase is responsible for transferring a group from one position to another position. So what they do is they just transfer the phosphate group from the 1 position to the 6th position, as simple as that. After that, the glucose 6-phosphate enters the glycolytic pathway. It is converted to pyruvate, then to acetyl-CoA, which enters the TCA cycle, and then results in the formation of energy. Are we clear about this? Yes or no? Are we clear about this? Okay. All right. Yes, UDP glucose to galactose by 4-epimerase. The last enzyme is phosphoglucomutase. Mutases are responsible for transferring a group from one position to another position. So phosphoglucomutase. Galactose 1-phosphate uridyl transferase. If you find this confusing, just remember the name, and you don't have to remember this part. Okay, you don't have to remember this part if you don't want to. This is not high yield. The name is high yield. Okay, lactose forms glucose and galactose. Right, lactose is broken down to glucose and galactose. Any other question? Okay, so this is the galactose metabolism. This is the galactose metabolism. Okay. Okay. Now, what happens over here is that this galactose, if it cannot convert or if it cannot go through this pathway, last piece of information. If galactose, if it is not converted to this pathway, for example, let's say galactokinase is missing. Okay. For example, there's no galactokinase. Then what happens is this galactose is converted to an osmotically active substance. This substance is known as galacticol. And the enzyme is also pretty high yield. This enzyme is called aldose reductase. This is the final piece of information from galactose metabolism. Normally, galacticol is not formed in normal amount. This is a pathology. Okay. Pathology, meaning that if anyone has galactokinase deficiency, then it won't go this way. It will go this way and result in the formation of galacticol. We'll talk about this in the future later. Okay. We're just talking about the physiology. We'll talk about the pathologies later. Are we clear about this? Okay. Now, can we talk about the fructose metabolism? Aldose reductase. Aldose reductase. Okay. Aldose reductase. Okay, now can we talk about the fructose metabolism? Fructose metabolism. This conversion in blood. Well, basically in the cells. Okay, in the cells. Okay. Can we begin with fructose metabolism, please? All right. Okay, before we begin with fructose metabolism, can I give you guys 1 minute to read this? I'll give you guys 2 minutes. Okay, read this for 2 minutes, and then after you guys have read this out, then I will move on to fructose metabolism. Okay.
Serious.
Okay, are we ready for the galactose metabolism? Yes or no? Are we ready for the galactose metabolism? Okay, so step by step, number one, lactose is broken down to which product? Lactose is broken down to which product? Glucose and galactose. Then galactose is converted to which product? Galactose is converted to which product? Galactose 1-phosphate. Which enzyme is responsible over here? Galactokinase. Next one, galactose 1-phosphate is converted to which product? Right. Which enzyme is responsible over here? Galactose 1-phosphate uridyl transferase. Very good. Now, glucose 1-phosphate is converted to which product? Glucose 6-phosphate. Which enzyme is responsible over here? Okay. Glucose 6-phosphate is converted to which product? Glucose 6-phosphate is converted to which product? Fructose 6-phosphate. Fructose 6-phosphate is converted to which product? Fructose 6-phosphate is converted to which product? Fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is converted to which product? Fast answers, please. Two products. You guys remember the two products? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then glyceraldehyde 3-phosphate is converted to which product? 1,3-bisphosphoglycerate, right? And then 1,3-bisphosphoglycerate is converted to which product? 3-phosphoglycerate, 2-phosphoglycerate, then phosphoenol pyruvate, and then pyruvate. Then pyruvate is converted to acetyl-CoA, converts, and then it is converted to citrate by oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, right? Succinyl-CoA, succinate, fumarate, malate, and then the cycle goes on. Okay, okay. I'm way faster than you guys. At least because I'm using my voice, you guys are using the chat box. But do you guys get the gist of how everything is being broken down over here? Okay. Okay. So can you guys be really confident that you guys know exactly how you are breaking down the things in your body step by step? Okay. Now, before we go for our 15 minutes break, can we do the fructose metabolism? It will take me 2 minutes. Okay, it will take us 2 minutes to learn about fructose metabolism. First of all, fructose. What is the pathway? I mean, what is the form of the? Okay, so my question is, so like, what is the source of fructose? Can we assume that the source of fructose is basically from fruit sugars, right? Fruit sugars. So from the fruits, we get sucrose. Sucrose. Sucrose, fruit sugar. Sucrose is broken down by sucrase to form fructose, and then what else? Glucose. Fructose and glucose. Okay, fructose is converted to fructose 1-phosphate. Can you guess which enzyme is working over here? Fast answers, please. Fructose 1-phosphate. Which enzyme is working over here? Kinase. Very good. The enzyme is fructokinase. Once again, ATP will be broken down to form ADP. Okay, fructose 1-phosphate. Fructose 1-phosphate over here is converted to it's converted to dihydroxyacetone phosphate. Dihydroxyacetone phosphate. And it's also converted to glyceraldehyde. Glyceraldehyde. Okay. Now, this dihydroxyacetone phosphate and glyceraldehyde, both of them, they are converted to glyceraldehyde 3-phosphate. If glyceraldehyde 3-phosphate is formed, then can we assume that they will enter the glycolytic pathway and be converted to 1,3-bisphosphoglycerate, then 3,2-bisphosphoryl pyruvate, acetyl-CoA? Yes or no? Yes or no? Okay. So this is the pathway of fructose metabolism. This conversion of fructose 1-phosphate to dihydroxyacetone phosphate, the name of this enzyme is very high yield. The name of this enzyme is aldolase. Do not confuse aldolase with aldose reductase. Aldose reductase will convert, it's an S. Aldose reductase will convert galactose to galacticol. Aldolase will convert fructose 1-phosphate to dihydroxyacetone phosphate. Okay, are we clear about this? Yes or no? Okay. If this reaction is not working over here, then fructose can directly be converted to fructose 6-phosphate. Fructose 6-phosphate. The enzyme is hexokinase. Okay. Once again, we get sucrose from the sugar. Sucrose is broken down by sucrase to fructose and glucose. Fructose is converted to fructose 1-phosphate by fructokinase. Fructose 1-phosphate is converted to dihydroxyacetone phosphate and glyceraldehyde by aldolase. And these two converge to glyceraldehyde 3-phosphate, and then this is converted to pyruvate, and acetyl-CoA is formed from the TCA cycle. But if this reaction does not work, then fructose has another pathway, that is this pathway, where fructose is converted to fructose 6-phosphate by the enzyme hexokinase. Okay, hexokinase. Are we clear? Hello? Okay. Are we clear about this, guys? Okay. So my question to you is, if you have galactokinase deficiency, do you have formation of an osmotically active substance? Yes or no? If you have galactokinase deficiency, do you have the formation of an osmotically active substance? What is the name of the enzyme that will convert galactose to that osmotically active substance? Aldose reductase. If you have fructokinase deficiency, do you have the formation of an osmotically active substance? No. Why? Because fructose is converted to which product? Fructose is converted to fructose 6-phosphate by the enzyme hexokinase. So if any of the pathways have to undergo, the formation of osmotically active substance and form cataract, which one will form cataract? Which one will form cataract? Galactose. Okay, are we clear about this now? Okay. So can I give you guys 1 minute to look at the sucrose pathway, the fructose pathway, and then we can move on and take our break and come back? Cataract. Basically, if you have galactokinase deficiency, then you have cataract. If you have fructokinase deficiency, then you do not have cataract. Because if you have galactokinase deficiency, then galactose is converted to galacticol by the enzyme aldose reductase. So that's that. Okay. Okay, so can I give you guys 1 minute to learn about the sucrose pathway, which we just talked about over here? Cataract. Basically, if you have fructokinase deficiency and galactokinase deficiency, which type of deficiency will produce cataract? That was my question. The answer to this is galactokinase deficiency will produce cataract. If you ask me why, then if galactokinase is not there, then galactose will be converted to galacticol by the enzyme aldose reductase. And galacticol is an osmotically active substance which will accumulate in different parts of the body, especially in the eyes. And osmotically active substance accumulation in the eyes can result in the formation of cataracts. Next one is fructose. On the other hand, if fructokinase is not working, then fructose will be converted to fructose 6-phosphate by the enzyme hexokinase, and this will enter the glycolytic pathway and will be broken down to form energy. So fructokinase deficiency will not produce cataract. This is a very high yield discussion, which we will have in the future. This is the pathology. Okay, so don't worry about this right now. If it's too much, we'll talk about this in the future. Okay. Okay, are we ready with the fructose metabolism? First of all, sucrose is converted to which two substance? Fast answers, please. Sucrose is converted to which two substances? Glucose and fructose. Then next one, fructose is converted to which one? Fructose is converted to which one? Good. What is the name of the enzyme? What is the name of the enzyme? Okay, next one. Fructose 1-phosphate is converted to which substance? What is the name of the enzyme over here? Aldolase. Very good. Aldolase. Okay. Aldolase. That's that. Okay. So next one is what is the function of aldose reductase? Aldose reductase. Formation of galacticol from galactose. What is the function of aldolase? Okay. Forms dihydroxyacetone phosphate. Okay. Are we clear about this? Yes or no? Question. Yes. What is your question? What is your question? Do you have a question, Dr. Karbasi? Aldolase. Aldolase will form both glyceraldehyde and dihydroxyacetone phosphate. And glyceraldehyde and dihydroxyacetone phosphate will both be converted to glyceraldehyde 3-phosphate. Is it clear now? Okay. Okay. Fructose 1-phosphate will be converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by aldolase. Is that over here? Fructose 1-phosphate is converted to dihydroxyacetone phosphate and glyceraldehyde phosphate by aldolase. Okay. Are we clear? Okay. Can we take that break right now, or do you want to take the break later? Okay, so after we come back, we will talk about some other pathways. Okay, I want to get done talking about all the pathways today, okay, because tomorrow is a new year, right? So I want to, okay, so let me tell you guys something. I want to get done talking about the pathways, because once we understand the pathways, then everything else over here will be extremely easy. Everything over here will be extremely easy. So I want to talk about all the pathways, urea cycle, this and that and everything. So after we are done learning the pathways, then we can talk about the diseases very quickly. So although it might take us some time to finish the pathways, but it's better for us so that we do, because from tomorrow onwards, we can talk about the diseases or the pathologies associated with this pathway. How does that sound? Does that sound okay? We'll try to cover as much as possible today, but our main focus for today is the pathways. Okay. Do we have class tomorrow? Yes. Is there any problem if we take tomorrow off? Yes, because if we take tomorrow off, then we will have an extra class. Already we have to take an extra class for Christmas. Do you guys remember? We have to take an extra class for Christmas, right? So that's that. Okay. So that's why we have to do the class tomorrow. I apologize, but if you guys want, I can keep the class short. Do you guys want me to keep the class short and then do extra timings later? Maybe, for example, instead tomorrow, instead of doing 5 hours, we can study for 2 hours and 3 hours and then add extra 1 hour later next week. How does that sound? No. Okay. So if it's a no, then we'll do the normal amount of classes. Okay. Okay. Okay, no need to take extra class for Christmas. The thing is, the way we charge you guys. Okay, so hear me out. The way we charge you guys is that we charge you guys $1 per hour, right? So if we charge you guys $1 per hour, and the total hours in a week is 25 hours. So if we do not take a makeup class for last week, then it's unfair for you guys, because you guys made a transaction for $25. So when you guys made the transaction for the $25, you guys paid for 25 hours. So last week, we only got to do 20 hours instead of 25 hours. So it's very unfair if on my counterpart, if I do not take extra class for Christmas, okay? Because if that was the case, then I should have mentioned that we would not have the class on Christmas, since it was a very spontaneous decision, and keeping everything under consideration, we had to make that decision out of nowhere. That's why I want to make sure that we are fair with you guys regarding the transactions and the hours. And that's why we feel the need to make up with an extra class. Okay, so that's what we're going to do. And about tomorrow, we can try to keep the class short. Okay, so let's say instead of 5 hours, we can keep the class short to 4 hours, and then obviously we will make up another hour later if needed. Okay. Next one. It's not about the money. Just tell us about schedule change ahead so as to use our day for something else. Yes, of course. We apologize for Christmas, to be honest. We were not sure about taking the holiday on Christmas because that was the only day that, yes, because that was the only day we canceled the class without having a prior discussion. Okay, but if you keep everything into consideration, then we have not canceled any classes except the one for Christmas. And since we did that, that's why we want to make it up. Okay, and 100% we will keep your daily activities into consideration next time when we make or next time when we plan on not doing the classes. Okay. So once again, we apologize for not doing the classes on Christmas, but in the future, when we say a class will not happen, it will not happen. Okay. Are we clear about everything? Do you guys have any other questions? Okay. So with that being said, can we take a break for 10 to 15 minutes and then come back and finish the cycles of biochemistry? Okay. Let's come back at 11:45 and let's finish the biochemistry cycles. Okay, is everyone back from their break? Is everyone back from their break? Everyone else, if you guys are back, can we get a yes on the chat box, please? How is the weather in your country right now? Is it winter over there in every one of your countries? Rainy. Yes. Okay. What's the lowest temperature that you guys have witnessed? What's the lowest of the temperatures? Dr. Karbasi, you guys have winter. Is it winter? It's winter in Germany. Okay, so what's the lowest temperature which you witnessed? 1. Okay. Okay. All right. It's cold in Texas. That is very unlikely for Texas. Okay. This year it wasn't really cold. Okay. Have you guys ever experienced New York cold weather? Have you guys ever been to New York or any other states in America? Chicago, yes. Okay, good. Okay, it can get very cold over here in New York. -9, -10. East does. Okay. Is everyone back from their break so that we can begin with the table? -3. Okay. Very good. I am so proud to have students from all over the country, and as a matter of fact, from all over the world as of right now, because this gives me an idea and confidence that if I were to visit any other country, I will know people in that country. If I were to visit your country and ask you for advices on places to visit, would you guys be able to tell me on which places I should go for tourism, for tours? Okay, then I look forward to it. Okay, if I plan on visiting any country, I'll see if I have students from that country or not, and then accordingly, I will see if you guys can give me some advices on places to visit in your country. Okay, good. So with that being said, hope you guys are back from the break. Hope you guys are liking the lecture today. We are going to finish the biochemistry pathways. Thank you so much, Dr. Naoud, Dr. Jordan, and everyone else. Okay, so let's begin with today's lecture. That is the first pathway, which we are going to begin right now, post-break, is fatty acid metabolism. Okay, fatty acid metabolism and fatty acid synthesis. But before we talk about that, we'll talk about fatty acid metabolism. Okay, first of all, if we have to talk about fatty acid synthesis, before we begin, is everyone ready? Do I have your attention? Do I have everyone's attention? Okay, good. Let's begin. So, first of all, the acetyl-CoA that was formed from the breakdown of pyruvate, we said that the acetyl-CoA will enter the TCA cycle in the mitochondria, right? Will enter the TCA cycle in the mitochondria to produce energy. But what happens to people who has a lot of carbohydrate and who does not work out or walk or do anything with the carbohydrate? For example, let's say you have a high carbohydrate dinner, okay? Dinner rich in high carbohydrate. For example, you have breads, rotis, and then biryanis and all these other things and rice and stuff, and then you go straight to bed. What would happen to the carbohydrate? Does anyone have a good answer for me? What would happen to the carbohydrate? Carbohydrate will be stored as fat. Very good. So what happens is that all the glucose which you have had will be broken down to pyruvate, and then the pyruvate will be converted to acetyl-CoA, and that acetyl-CoA, instead of entering the TCA cycle for the production of energy, it will enter the fatty acid synthetic pathway, fatty acid synthesis pathway. So right now, let's say you have a high carbohydrate meal, and then you go to bed without working out or doing anything or walking or anything else. That acetyl-CoA will be converted to malonyl CoA. And the enzyme that is responsible for conversion of acetyl-CoA to malonyl-CoA is acetyl-CoA carboxylase. Basically, a carboxyl group is added. So if we talk about carboxylase, which cofactor is being used in this pathway? Which cofactor or which vitamin is used in this pathway? Fast answers, please. Carboxylation is by B7. Very good. Okay. Really glad to see you guys very confident about the water-soluble vitamins and then which pathways they're used. So B7 is biotin. Biotin is used for all types of carboxylation reactions. Then malonyl-CoA is converted to fatty acid. Okay, malonyl-CoA is converted to fatty acids. Fatty acid, and the enzyme that is working over here is fatty acid synthase. Okay, and then this fatty acid will convert with glycerol to form fat. Are we clear about this? Form fats and lipids. Okay, are we clear about this? Yes or no? Okay, good. Now, this fatty acid will convert again with glycerol to form lipids, and that lipid will be stored in the adipose tissue, and that's how we get fat from having excess amount of carbohydrates at night or having a lot of carbohydrates without having to work out or do anything. So that's that. Okay, now this fatty acid, instead of converting to now lipid, if we have to talk about the breakdown of fatty acid, now let's talk about the breakdown of fatty acid. If fatty acid is broken down, the fatty acid will convert, instead of converting to lipid, the fatty acid will now be converted. Now we're talking about the breakdown, okay? We're talking about breakdown of fatty acid, okay? Now, breakdown of fatty acid. The fatty acid, what would happen is, instead of converting to lipid, the fatty acid will be converted to acyl-CoA. Not acetyl-CoA, it will be converted to acyl-CoA. The enzyme that is working over here for conversion of fatty acid to acyl-CoA is a very simple enzyme. That is acyl-CoA synthase. Acyl-CoA synthase. Acyl-CoA synthase, once it is formed, it enters a very high yield cycle. Okay, first of all, fatty acid oxidization. Which organelle is responsible for fatty acid oxidization? Mitochondria. So what is our goal? What is our goal? Our goal is we want to make sure that the acyl-CoA is successfully entered into the mitochondria. Yes or no? Yes or no? Yes. Okay, good. In order for this one to happen, so right now this acyl-CoA, this is in the cytoplasm. our goal is we have to make sure this enters the mitochondria for oxidation. So a very high yield pathway over here is this pathway is known as the carnitine shuttle. How many of us have heard of carnitine shuttles? Carnitine shuttle. Have you guys heard of carnitine shuttle? Okay. Okay. As a matter of fact, carnitine, a lot of nowadays, a lot of newer supplements in bodybuilding, they sell carnitine because carnitine is used for transferring excess S-alcohol to the mitochondria. And once that does, the carnitine, it is responsible for fat breakdown. So for a lot of fat burners, you have a lot of fat burners which has a lot of carnitine inside them. And that is actually a USMLE step 1 question. That's why I'm talking about fat burners, because they are used over here widely in USA. So you have carnitine. Carnitine, if you think about it, it's more or less like a car, which was going on its pathway, right? And then it saw the acyl-CoA, it picked up the acyl-CoA, and with the acyl-CoA now in the car, this is known as acyl-carnitine. 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 This acyl-carnitine is easily transportable across the mitochondrial matrix over here, and it enters the mitochondrial matrix because acyl-CoA cannot enter the mitochondrial matrix. That's why carnitine converted acyl-CoA to acyl-carnitine. Now this entered the mitochondrial matrix. Now this is known as acyl-carnitine. Acyl-carnitine. And the enzyme that is working over here is known as carnitine acyl transferase 1. So as you would like to see, or you would like to imagine that there was a car with a driver who loves cat. When the car was passing by, the car saw acyl-CoA standing over there, asking him, where do you want to go? Acyl-CoA said, I want to enter the mitochondria. The carnitine picked up the acyl-CoA with the help of the cat. It converted acyl-CoA to acyl-carnitine. Acyl-carnitine successfully entered the mitochondria, and now it's time for the car to leave and go back to the cytoplasm. How will that happen? Is that acyl-carnitine will be converted back to carnitine by CAT2, carnitine acyl transferase 2. And carnitine will now be will now be transferred from the mitochondria to the cytosol once again. This shuttle is known as carnitine shuttle. Are we clear about this? Cytoplasm, mitochondria. Are we clear about this shuttle? Yes or no? Are we clear about this shuttle? Yes or no? Okay. Okay. Now the thing is, after acyl-carnitine is converted to carnitine, this acyl-carnitine, what would happen is while this process is going on, this will convert this acyl-carnitine to acetyl-CoA once again. Acetyl-CoA. Because what is our goal for breaking down fat? Okay. What is our goal for breaking down fat? We're talking about fat breakdown. Can we say that we break down fat to produce energy? Yes or no? Can we break down fat to produce energy? Yes. So this is the mechanism by which fat is broken down. So fat is broken down to form acetyl-CoA once again, and this acetyl-CoA will enter the TCA cycle and then result in the formation of energy, right? And this is the pathway of fat breakdown. What happens over here is that you have another enzyme. This is acyl-CoA dehydrogenase. Okay, and this breakdown of acyl carnitine to acetyl CoA by the enzyme acyl CoA dehydrogenase, this results in the formation of two types of products, FADH and NADH. This is the whole mechanism of fat breakdown. Are we clear about this? Yes or no? Okay, so what's happening over here? Let me repeat. First of all, acetyl-CoA. Okay, first of all, you have acetyl-CoA, which is coming from excessive carbohydrate. And if we take excessive carbohydrate, this acetyl-CoA is converted to malonyl-CoA by the enzyme acetyl-CoA carboxylase. This malonyl-CoA is converted to fatty acid by fatty acid synthase, and then fatty acid is converted to lipids when they combine with glycerol. So this is the fatty acid or lipid synthesis. This is the lipid synthetic pathway. Now, if we talk about breakdown, that is in terms of, let's say, in a period of fasting where we need excess energy to function, we have fats which are stored in the adipose tissues, which can be broken down to produce energy, right? So this fat is broken down to acyl-CoA. This fatty acid is broken down to acyl-CoA by acyl-CoA synthase. Acyl-CoA cannot enter the mitochondria. So in order for acyl-CoA to enter the mitochondria, they enter a car called carnitine with a CAT, carnitine acyl transferase. Excuse me. And this is converted to acyl carnitine. This acyl carnitine can now be transferred from the cytoplasm to the mitochondria. And once it enters into the mitochondria, it's time for the car to leave. So carnitine is formed from acyl carnitine once again by carnitine acyl transferase 2. And now this carnitine can go back. And this shuttle keeps on happening throughout the whole day to make sure that there is proper fat transport inside the mitochondria. Now, if after this happens, through this reaction, you have another enzyme called acyl-CoA dehydrogenase, which converts acyl carnitine to acetyl-CoA. And this acetyl-CoA enters the TCA cycle in the mitochondria to produce energy. While this is produced, this pathway results in the formation of two byproducts, that is FADH and NADH, as simple as that. This is your fatty acid metabolism. Okay, are we clear about this? Are we clear about this? Okay. So can I give you guys 1 minute before we move on to the next pathway? Okay, I will give you guys 1 minute till we move on to the next pathway. In that 1 minute, please read the pathways and then I'll ask you questions. It's 12:03. I'll ask you the question at exactly 12:05. Okay, let's do this. Okay, are you guys ready? Okay, so fatty acid synthesis. What is the first substrate of fatty acid synthesis? Fast answers, please. What is the first substrate of fatty acid synthesis? Malonyl-CoA. Then malonyl-CoA is converted to which one? Fatty acid. Very good. Fatty acid converts with glycerol to form lipids. That's that. If you want to break down fatty acid, then fatty acid is converted to which one? Acyl-CoA, right? Right? Acyl-CoA synthetase. And then acyl-CoA, can it enter mitochondria? Yes or no? Can it enter the mitochondria? Acyl-CoA? No. How will it enter the mitochondria? How will it enter the mitochondria? Carnitine shuttle. Very good. So it enters the mitochondria as acyl-carnitine, and then it's converted to acetyl-CoA by acetyl-CoA dehydrogenase. Have you guys understood this step? What are the two byproducts of acetyl-CoA dehydrogenase? What are the two byproducts of acetyl-CoA dehydrogenase? FADH and NADH. Okay. Very good. Okay, next one. Okay. Next one that I want to talk about is, so we talked about carbohydrate, we talked about fatty acid. Now can we talk about amino acid metabolism? Amino acid metabolism. Amino acid metabolism. Okay, I need you guys to put your attention to amino acid metabolism, because out of all the pathways which we have just studied, amino acid metabolism is the most high yield pathways. The amount of questions you will receive from amino acid metabolism is 5 times more likely than the questions you will receive from glycolytic or fatty acid pathways. Okay, so are we ready to begin amino acid breakdowns? Yes or no? Yes or no? Are we ready to begin amino acid breakdown? Guys, can I get a yes? Okay, good. Let's begin. So when we talk about amino acid breakdown or amino acid breakdown, the first amino acid that we are going to consider for this type of breakdown is, even all the charts, they focus on one amino acid especially. That amino acid is known as phenylalanine. Phenylalanine. So we're going to talk about phenylalanine metabolism. This is very high yield. And you guys will know exactly why this is very high yield. Okay. Phenylalanine metabolism. Now, phenylalanine is converted to another type of amino acid. Does anyone know this amino acid? It starts with a T. Anyone? Anyone? Tyrosine. Very good. Tyrosine. Okay, tyrosine. Okay. The enzyme that is working over here, the name of this enzyme is phenylalanine hydroxylase. Phenylalanine hydroxylase. This will convert phenylalanine to tyrosine. And there's another co-pathway that will work alongside phenylalanine, is that this pathway that is working over here, this pathway will be converting BH2 to BH4. BH is basically biopterine or biopterine. Okay, biopterine. And the enzyme is dihydrobiopterine reductase. This is the complexity of this pathway. Okay, biopterine, BH2 to BH4, and the enzyme is dihydrobiopterine reductase. Okay, now tyrosine will be converted to, okay, tyrosine will be converted to another substrate. This substrate is known as, does anyone know the name of this substrate? Anyone? Okay. Tyrosine will be converted to DOPA. DOPA. Okay. And in order for this pathway to happen, in order for tyrosine to be converted to DOPA, this cycle will also work in this way. Basically, there's another cycle happening over here. Okay? There's another cycle happening over here that will convert tyrosine to DOPA. And this cycle, once again, is BIOCHEMISTRY cycle. BH2 is converted to BH4. Okay, dihydroxy biopterine reductase. DOPA. Now, this DOPA that is formed over here, does anyone know what we get from DOPA? What do we get from DOPA? Anyone? What do we get from DOPA? Dopamine and norepinephrine, right? So DOPA will be converted to dopamine. Do you guys know the name of vitamin that works over here to convert DOPA to dopamine? We talked about this yesterday. Vitamin C, very good. Vitamin C, very good. DOPA decarboxylase. DOPA decarboxylase, and over here, B6. I think it's vitamin C, no? It's vitamin C, right? Not sure about this. Let me go see. Okay, 1 second. Let me go see about that. Okay. 1 second. 1 second. Okay, B6. You're right. Next step is vitamin C. Okay, good, very good. Dr. Iman, you are right. Dr. Hussam, you're wrong, and I am wrong too. Okay. Okay. So let's go back. Okay, let's go back. So over here, it's vitamin B6. My apologies. I am sorry. It's dopamine. Okay. Are we clear up to now? Are we clear up to now? Yes or no? Okay. Thank you, Dr. Iman, for correcting us. And okay. Next one, dopamine. Dopamine will be converted. The reason why I make mistakes, I make a lot of mistakes at times, it's because I'm trying to figure everything out from my knowledge. Okay, so my apologies for at times, and thank you so much for correcting me. Okay, dopamine will be converted to norepinephrine. Okay, norepinephrine. Now this step requires vitamin C. Okay, this step requires vitamin C, ascorbic acid. And norepinephrine will be converted to epinephrine. Okay, epinephrine. And the enzyme that is over here is phenylethanolamine N-methyl transferase. Very big enzyme. In short, we write PNMT. Phenyl and ethylamine N-methyltransferase. Phenyl ethanolamine N-methyltransferase. In short, we write PNMTA. Is this the first time you guys have heard of phenyl ethanolamine N-methyltransferase? Yes or no? Okay, I'm pretty sure you guys have heard about this enzyme before, right? Am I correct? Okay, good, very good. So phenyl ethanolamine N-methyltransferase. That is the name of the enzyme. Okay, this is amino acid metabolism. This is the amino acid metabolism. Now, what happens over here is if the tyrosine, which is converted to DOPA, this tyrosine has another pathway over here. If we just focus on tyrosine metabolism, tyrosine can be converted to either this pathway, or tyrosine is converted to another substance that is known as homogentisate. Homogentisate. Homogentisate is converted to, do you guys understand how in gluconeogenesis, when we talk about gluconeogenesis, do we talk about the fact that glucose is formed from fat and protein? Yes or no? Because the brain needs glucose, right? Okay. So if in order for this to form glucose, one of the substrate has to be formed that can enter the TCA cycle. Because if you have formation of the TCA cycle, then from the TCA cycle, you will have the pathway to go back to the formation of glucose, okay? And we'll talk about gluconeogenesis in a while. So once again, over here, tyrosine can be formed and transformed to DOPA, or tyrosine can be converted to homogentisate. Homogentisate can be converted to melyl acetoacetate. Melyl acetoacetate. The enzyme that is working over here is known as homogentisate dehydrogenase. This is a very high yield enzyme, homogentisate dehydrogenase. Okay, homogentisate dehydrogenase enzyme. Does anyone know the name of the disease if we have homogentisate dehydrogenase enzyme deficiency? Homogentisate. Alkaptonuria. Very good. Alkaptonuria. Homogentisate deoxygenase. My apologies. Homogentisate deoxygenase. I'm sorry. Not dehydrogenase, deoxidase. Homogentisate deoxidase. Okay. Homogentisate deoxidase. And this will result in the formation of melyl acetoacetate. Now, this melyl acetoacetate, this will be converted to, does anyone know what melyl acetoacetate will be converted to? Fumarate. Very good. This will be converted to fumarate. This is the amino acid pathway, amino acid breakdown pathway. And then the fumarate will enter the gluconeogenic pathway. Are we clear about this? Yes or no? So how many mistakes did I make in the phenylalanine breakdown pathway? 4 mistakes. How many mistakes did I make? I made 2 mistakes. Very good. I made 2 mistakes. What are the mistakes I made? It's for my own future reference so that I don't make the mistakes again. This is the one mistake I made, vitamin C. So please don't forget this, because I have read this pathway so many times. Still, I make mistakes because this is a very complex pathway. Another mistake I made was over here. Okay. 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 So once again, phenylalanine is converted to tyrosine by phenylalanine hydroxylase. Tyrosine is converted to DOPA by the help of the enzyme. What is the name of the enzyme over here? Tyrosine is converted to DOPA by dihydrobiopterin reductase, and then DOPA is converted to dopamine by Dopa decarboxylase, B6. Dopamine is converted to norepinephrine by vitamin C as a cofactor. Norepinephrine is converted to epinephrine as phenyl and ethanol amine and methyl transferase. And this is one pathway of tyrosine. Tyrosine can be converted to another pathway, to homogentisate. Homogentisate is converted to melyl acetoacetate. Melyl acetoacetate is converted to fumarate, and fumarate is converted to malate, and then it enters the gluconeogenetic pathway. Okay, very, very important pathway. Can I give you guys 2 minutes to read this pathway once again before we move forward to the last pathway, that is urea cycle? Okay. The purple circle, which is BH2 to BH4, is biopterine, which is converted to BH4, BH2. This is done by the enzyme dihydrobiopterine reductase, which reduces BH2 to BH4, and this results in the formation of another co-pathway, which helps for the proper conversion of phenylalanine to tyrosine. And this occurs in 2 stages, over here and over here. That's why. Okay. Is everyone clear about this? Okay. Now, can I give you, okay, why not TCA cycle? I mean, fumarate. Why not TCA cycle? Amino acid does not result in the direct formation of acetyl-CoA. Amino acid results in the direct formation. Amino acid is broken down to enter the gluconeogenetic cycle to form glucose first, and then that glucose is converted to acetyl-CoA, and then it enters the TCA cycle. Okay, that's why. Okay, are we clear? Fumarate is a part of the TCA cycle, yes. Do you remember where fumarate was there? Yes. Yes, it goes to the TCA cycle, but it enters the gluconeogenetic pathway. It will produce some amount of enzyme, I mean, some amount of energy, but it will produce the most amount of energy after the fumarate is converted to glucose by the gluconeogenetic pathway, and then that is converted to acetyl-CoA, and then it enters the TCA cycle. Okay? Okay. So can I give you guys 2 minutes to read this pathway before we move forward? Okay, is everyone ready to talk about phenylalanine pathway? Is everyone ready to talk about the phenylalanine pathway? First of all, phenylalanine is converted to which amino acid? Fast answers, please. Tyrosine. What is the name of the enzyme? What is the name of the enzyme? Good. Then tyrosine is converted. Let's talk about the dopamine pathway first. Tyrosine is converted to which one? Fast answers, please. Okay. And then dopa is converted to which one? Dopamine. Okay. Dopa is converted to dopa is converted to dopamine. And then dopamine is converted to which one? Norepinephrine. Okay, good. Then another one is tyrosine metabolism. Tyrosine, if we want to make glucose from tyrosine, which pathway do we use? Okay, gluconeogenic pathway. So it's converted to homogentisate by homogentic acid deoxidase. Okay, and then homogentisate is converted to mallyl acetoacetate. Mallyl acetoacetate is converted to fumarate. Are we clear about this? Are we clear about this? Okay, now let's talk about the last pathway that for today that we want to discuss. The last pathway that we want to discuss for today is, does anyone know? We have another cycle. We drew another cycle. What pathway could this be? Anyone? Urea cycle. Very good. Urea cycle. Okay, this is the urea cycle. Okay. Urea cycle is basically the cycle that is used for proper excretion of ammonia. Am I correct? Ammonia, right? What is ammonia? What is ammonia? Ammonium. Ammonia. What is ammonia? It's a waste product, right? What would happen if ammonia, if there's high ammonia and if it's not, if the urea cycle is not working, can we have excessive ammonia that can build up in the blood and result in hepatic and cerebral encephalopathies or right? And encephalopathy. Can patients pass away and die from encephalopathies? Yes or no? Yes. Okay. So we have to do the urea cycle. Urea is the urea is the what can I say? Urea is the less toxic compound of ammonia. It's equally toxic, but urea can be easily it could be easily getting rid of the body. I mean, you can easily get rid of urea. But in order for urea to be formed, this ammonia has to be converted to urea, and urea is less toxic. That's why we have this cycle over here, which is known as the urea cycle. Known as the urea cycle. Okay. Now, first of all, ammonia is the main problem. Ammonia, when it is produced, ammonia combines with carbon dioxide, which is another byproduct, carbon dioxide, to form carbamyl phosphate. Carbamyl phosphate. Carbamyl phosphate. Ammonia converts to carbamyl phosphate after it binds with carbon dioxide. This process is done in the presence of an enzyme called carbamyl phosphate synthase 1. Do you guys remember carbamyl phosphate synthase 2? Where was carbamyl phosphate synthase 2 used in? Very good. Where does carbamyl phosphate synthase 2 work? In which organelle? And in which organelle does carbamyl phosphate synthase 1 work? Synthase 1 in mitochondria, 2 in cytosol. Cytosol, okay? So carbamyl phosphate synthase 1 converts ammonia to carbonyl phosphate after it binds with carbon dioxide in the presence of ATP. Okay, in the presence of ATP. They use up ATP and they result in the formation of carbonyl phosphate. Carbonyl phosphate enters the urea cycle by forming one of the substrate of the urea cycle. This substrate is known as citrulline. Citrulline. And the way carbonyl phosphate forms citrulline is that carbonyl phosphate combines with ornithine. Carbonyl phosphate and ornithine, they bind together to form citrulline. Okay. Citrulline is converted to another substrate that is called arginosuccinate. Arginosuccinate. And the enzyme that works over here, this enzyme is known as arginosuccinate synthase. And in order for citrulline to be converted to arginosuccinate, they combine with another amino acid from the outside. This amino acid is known as aspartate. Aspartate. Okay. So before we move forward, let's do a recap so that we're not overwhelmed with the knowledge. We're talking about the urea cycle. It's important for the excretion of ammonia. Ammonia combines with carbon dioxide in the presence of carbonyl phosphate synthase 1 in the mitochondria. With the help of ATP, it forms carbonyl phosphate. Carbonyl phosphate combines with ornithine to form citrulline. The enzyme that is working over here is called ornithine transcarbamylase. Very high yield enzyme. This enzyme is called ornithine transcarbamylase. Ornithine transcarbamylase converts ornithine and carbonyl phosphate to citrulline. Citrulline combines with aspartate in the presence of arginosuccinate to form, in the presence of arginosuccinate synthase to form arginosuccinate. Arginosuccinate is converted to arginine. Arginosuccinate is converted to arginine by the help of the enzyme called arginosuccinate lyase. Meaning that they break down the bond in between the arginosuccinate and this results in the formation of an amino acid called arginine. This arginine that is formed over here, this results in the formation of ornithine. How? By the help of an enzyme called arginase. Easy because it goes by the same name as arginine. So the help of the enzyme called arginase. Okay, it is by the help of arginase that you have the formation of urea. And this urea is formed because when this reaction is taking place, water is also used, and this dilutes the reaction to result in the formation of your urea. Then this urea is excreted. Okay. Okay, this is the urea cycle. Are we clear about this? Yes or no? Are we clear about this? Are we clear about this, guys? Okay. Okay. Now, I'll go with the urea cycle again step by step because I know this is very complex. So let's begin from over here. Ammonia combines with carbon dioxide in the presence of carbonyl phosphate synthase 1 and ATP to form carbonyl phosphate. Carbonyl phosphate combines with ornithine to form citrulline in the presence of ornithine transcarbamylase. Citrulline is converted to arginosuccinate by arginosuccinate synthase when it combines with aspartate. Arginosuccinate is converted to arginine by arginosuccinate lyase. Arginine is converted to ornithine by the help of the enzyme arginase. And in this reaction, when arginase is breaking down arginine, it results in the formation of urea. And water also helps because water helps to dilute the byproduct. And this diluted product is called urea. This urea is then excreted from the body via the urine, the kidney. Okay? Are we clear about this? Yes or no? Okay. So can I give you guys another, how long? Can I give you guys 2 minutes to read this? Okay. I'll give you guys 2 minutes. I'll give you guys 2 minutes. Okay. Urea cycle. Arginase breaks down arginine to form ornithine. And in this breaking down of arginine to ornithine, you have the production of a byproduct called urea when water dilutes the byproduct. This byproduct, when it's coming, water dilutes it and forms urea. That's that. Okay. This is known as the urea cycle. Okay, I'll ask you guys the question at 12:40. Exactly at 12:40, I'll ask you guys the question. Okay. Now, let's talk about the urea cycle. Okay, let's talk about urea cycle. First of all, what is the first reaction of urea cycle formation? What is the first reaction of urea cycle formation? Ammonia plus carbon dioxide in the presence of carbonyl phosphate synthase 1 converts ammonia to carbonyl phosphate. Carbonyl phosphate enters the urea cycle via reaction of which substrate of the urea cycle? Citrulline. Forms citrulline when it combines with ornithine. What is the name of the enzyme over here? Ornithine transcarbamylase. Very good. And then citrulline is converted to arginosuccinate by which enzyme over here? Arginosuccinate synthase when it combines with aspartate. Very good. Arginosuccinate is converted to which one? Arginosuccinate is converted to arginine. Arginine is converted to which one? The last step, the most important step. Okay. Urea. There we go. Okay. So with that being said, we have finished all of the major pathways of biochemistry. And I know that a lot more is left over here, but we are done with the major pathways. Everything over here, we're done with the major pathways. Glucose, galactose, this one. Oh, gluconeogenesis. Let's talk a little bit about gluconeogenesis. The gluconeogenetic pathway, okay, is the same as glycolytic pathway, except that the oxaloacetate that is formed from the TCA cycle, it enters the gluconeogenetic pathway. So the first substrate is oxaloacetate. It enters the gluconeogenetic pathway when it is converted to pyruvate in the presence of pyruvate carboxylase. Pyruvate carboxylase. And also in the phosphoenol pyruvate by phosphoenol pyruvate carboxylase. So these two are the two pathways by which oxaloacetate forms phosphoenol pyruvate, and then everything starts going this way, the opposite. So before we talked about how 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. For example, right now, if it goes upwards, that we'll talk about 3-phosphoglycerate being converted to 1,3-bisphosphoglycerate, and everything going upwards like this. Okay. So this is the gluconeogenetic pathway. Okay. Okay, it will go one way. Are we clear about this? Yes or no? Are we clear about gluconeogenesis? We're not clear about gluconeogenesis. Gluconeogenesis. Okay, let's talk about gluconeogenesis. Okay, gluconeogenesis. Let me break it down for you. Give me 1 second. Let me break it down for you. Okay. Okay. So what's happening over here is that you have the oxaloacetate, right? You have oxaloacetate. Oxaloacetate will be converted to malate. Are we clear? Oxaloacetate will be converted to malate. Okay. Malate, oxaloacetate, which is converted to malate. Do you guys remember in TCA cycle, we talked about the fact how malate is converted to oxaloacetate? Yes or no? Do you guys remember in TCA cycle, we talked about how malate is converted to oxaloacetate, right? But for gluconeogenesis to occur, the oxaloacetate will be converted to malate. Okay, and the enzyme, once again, is the same enzyme, that is malate dehydrogenase. Malate dehydrogenase. So when malate is formed once again from the TCA cycle, this malate will be converted back to oxaloacetate once again. Oxaloacetate, because it will take the oxaloacetate away from the TCA cycle. This cycle where oxaloacetate was used, oxaloacetate will be taken away from the TCA cycle and formed into oxaloacetate from the malate once again. This oxaloacetate will be converted to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase. And then this phosphoenol pyruvate will be converted to 2-phosphoglycerate, right? 2-phosphoglycerate. And then from 2-phosphoglycerate, you will have 3-phosphoglycerate. From 3-phosphoglycerate, you will have 1,3-bisphosphoglycerate, and so far and so forth till it reaches to the formation of glucose. Are we clear about this? This is the gluconeogenesis pathway. Okay. Okay. Now we're not done over here. Do you know why we're not done? Okay. Do you know why we're not done? We're not done because over here in gluconeogenesis pathway, we have to talk about this enzyme. I mean, we have to talk about this enzyme, and we have to talk about this enzyme. Basically, from when fructose 6-phosphate was converted to fructose 1,6-bisphosphate, we talked about phosphofructokinase. But when fructose 1,6-bisphosphate will be converted to fructose 6-phosphate, the enzyme that will work is known as fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. And when it will go upward from fructose 6-phosphate, it will convert to glucose 6-phosphate. And glucose 6-phosphate, instead of glucose being broken down by glucokinase or hexokinase to glucose 6-phosphate, glucose 6-phosphate will be converted to glucose by the help of an enzyme, which is known as glucose 6-phosphatase. Glucose 6-phosphatase. This is the only difference, that is the difference of enzymes in the glycolytic and gluconeogenetic pathways. Okay? Are we clear about this? Is this the first time you guys heard about this information, yes or no? Or did you guys know about this from before? Did you guys know about this from before? Can we get some fast answers, please? Okay, again. After 3-phosphoglycerate, you will have formation of 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate will be formed into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate will form fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate will be converted to fructose 6-phosphate by fructose 1,6-bisphosphatase. This will be converted to glucose 6-phosphate, and glucose 6-phosphate will be converted to glucose by glucose 6-phosphatase. That's that. Okay. That is the gluconeogenetic pathway. Okay, are we clear about this? Everyone, are we clear about this? Okay. Out of these pathways, which two pathway did we not discuss? This pathway and this pathway. Okay. This pathway, I will discuss this pathway when we study glycogen storage diseases. So I will not discuss this right now. I will discuss this when we study glycogen storage diseases. Okay. And this pathway, I will talk about in the next class, not right now. Okay. I'm pretty sure you guys know ketogenesis, but I'll talk about this in the next class. Okay. Are we clear about this? Yes or no? Okay. HMP shunt pathway. HMP shunt pathway, I'll discuss about this tomorrow. Sorry. Wait, I'm sorry. 1 second. HMP shunt pathway. HMP shunt pathway. Where is HMP shunt pathway? HMP shunt pathway. I'll discuss this tomorrow when we come to HMP shunt. Okay. Okay. Are we clear about this? Okay. Since we are a little bit, so the reason why I don't want to discuss any more pathways is because our concentrations are being broken down more or less, because we have talked about so many pathways. And to be honest, I also want to take a break from talking about the pathways because I can't concentrate as much. And to be honest, from what looks like I'm making some mistakes, and I want to stop over here, and I want to start with the pathways from tomorrow once again. But we don't have any more pathways left. We just have 2 more pathways. That is HMP shunt pathway, which is not that high yield because you will not be asked direct questions about this. So we'll talk about the HMP shunt pathway tomorrow. And but there are some things I want to talk about. Okay, I was thinking if it was possible to talk about lysosomal storage diseases. Are you guys interested to talk about lysosomal storage diseases? Yes or no? Okay, I really want to talk about lysosomal storage diseases. This is very high yield. And I want to also talk about glycogen storage diseases. Okay, so the reason why I want to talk about glycogen storage disease and lysosomal storage diseases is because if you have to receive questions from biochemistry, these are the 2 tables from which you will receive questions from biochemistry over and over and over again. And the way we will talk about glycogen storage disease and lysosomal storage disease is we will look at the picture mnemonics of these things from physio, learn about them, and then we will talk about these diseases in details. Okay, so do you guys want to do this today? Yes or no? Okay, can we at least do one? Let's say lysosomal storage diseases instead of glycogen storage diseases. 1, can we at least do 1? Break first. Okay, how long do you guys want to take the break? Okay, 5 to 10 minutes. 5 to 10 minutes. Okay. Okay, how about this? How about, since lysosomal storage diseases, they're very high yield, do you guys want to start the discussion tomorrow, first thing in the morning, with lysosomal storage disease and glycogen storage disease? Because that way it will remain fresh in the mind. Because right now you guys are so, I feel like you guys are not feeling it. You guys are not that confident, or right now after pathways it could be a bit difficult. I completely understand. After after reading so many pathways, you guys could be worn out. Okay, so how about this? How about for today we take a break from first aid and the first thing that we do tomorrow is we start with glycogen storage disease and lysosomal storage diseases early in the morning, and then we move on with the rest of the biochemistry diseases. Okay, and for now, let's take a break and let's come back. And how about we solve some UWorld questions, because it's been a while since we did some UWorld questions. I mean, the UWorld notes. UWorld notes. Okay. Okay. All right. So let's take a break for 5 to 10 minutes. Let's come back at 1:05 and let's see what's up. Okay, is everyone back from their break? Can you guys hear my voice? Okay, do you guys have any plans for New Year's Eve, or is it already the 1st of Jan in some countries? Okay, good. Make sure you guys stay home with your family, stay safe, because coronavirus is still on the rise. So that's that. Reading the pathways. I would say that's the best way to spend your New Year's. Read the pathways of biochemistry. Okay, very good. Okay. Everyone else, do you guys have any plans for your New Year's? Can you guys hear my voice? If you guys can hear my voice, can I get a yes, please, on the chat box? Okay. Okay. So let's begin with our UWorld notes. Is everyone ready with the discussion on UWorld notes for the high yield UWorld notes, which are bit difficult, meaning that the percentage of the student who gets these rights are 30 to 40%, and this will give you a better edge to do well in your exam by going through them in the last dedicated periods. Okay, let's begin. Okay, if we have a patient with heart failure, please use the chat box to mention heart failure with muscle weakness. And if I tell you it's a type of muscular dystrophy, which type of muscular dystrophy is this? Anyone? Why do you guys think it's Duchenne's muscular dystrophy? Why not Becker's? Why not Becker's? Duchenne's more severe, that's correct, but Becker's is less severe. Right. But this patient has heart failure, muscle weakness without Gower sign. This patient did not have any Gower sign, but this patient had heart failure with muscle weakness. And for this question, the absence of Gower sign resulted in Becker's muscular dystrophy. Okay. Next one. Next one is macular adherence. What is another name for macular adherence? Does anyone remember the gap junctions, macular adherence? No. Another name for macular adherence is known as gap junction. Okay. For some reason, this was a high yield question. Next one. Gram-positive bacteria, catalase positive, and non-fermenting. What is this organism? What is this organism? Gram-positive, catalase positive, non-fermenting. Anyone? Non-fermenting mannitol. Anyone? What is this organism? Staph epidermidis. Okay. Next one. Okay. Osteonecrosis of the jaw. Which medication can cause osteonecrosis of the jaw? Anyone? Very good. Okay. Lipid-rich, protein-rich fluid in interstitial space. What is this fluid? Lymph. Very good. This is lymph. Okay. Severe combined immunodeficiency syndrome is caused by two types of, there are two reasons behind SCID. One is adenosine deaminase deficiency. Another one is interleukin IL-2R defect. Okay. In a patient with benign prostatic hyperplasia and hypertension, which drug can prevent both conditions at the same time? Which group of drug can prevent both conditions at the same time? Alpha 1 blocker. Very good. For example, tamsulosin, terazosin. Okay. mRNA synthesis mRNA synthesis my apologies, 1 second. Okay. mRNA synthesis can be blocked by, can you guys name some RNAs which can block mRNA synthesis? Which RNAs will block mRNA synthesis or mRNA transcription? MicroRNA and siRNA. Now, if I talk about drugs, can you guys tell me which toxin can prevent mRNA synthesis? Which toxin can prevent mRNA synthesis? Very good. It's called alpha amanitin. Alpha amanitin. Which organism produces alpha amanitin? Fungi. What is the name of the fungi? Amanita phyllodes. Okay. Or mushroom. Crazy mushrooms. Okay. Failure to thrive, hypotonia, hypertrophic cardiomyopathy with normal glucose. What is the diagnosis? Pompe's disease. We'll talk about this tomorrow, early morning, Pompe's disease. Okay. Most common location of anal fissure. What is the most common location of anal fissure? posterior midline of anal canal, distal to pectinate line. Okay, unilateral headache with autonomic sign symptoms. What is the type of headache? Autonomic sign symptoms. Cluster headache. Very good. Treatment of cluster headache. Triptans. Okay, triptans. Very good. Okay. Most common cause of aseptic meningitis. Aseptic meningitis, most common cause. Fast answers, please. Which one? Ecovirus. If you have to be very specific, it's ecovirus. Okay. Okay. Next one is lab procedure to detect mRNA. Which one? Fast answers. Very good. Okay. Most common cause of mediastinal midline mass. Anyone? Anyone? Midline mediastinal mass. Mediastinum. Thymoma. Very good. Treatment of respiratory syncytial virus. Specific treatment. A specific treatment. It's a monoclonal antibody. Does anyone know the treatment? Anyone? We'll talk about this. Okay. Next one. Okay. Painless generalized lymphadenopathy with splenomegaly with age above 65 years. Well, what is your diagnosis? CML or CLL? Which one? CML or CLL? Why do you think it's CML and why do you think it's CLL? First of all, tell me, why do you think it's CML? The answer is CLL. Okay. It's not CML. Why this is not CML? CLL, once again, is more common in patients above 65 years of age, and generalized lymphadenopathy is more common in CLL. Okay. And antiarrhythmic drugs that can prolong QT and QRS. What are the names of the drugs? QT prolongation, and QRS prolongation. Fast answers, please. Class 1A specifically. Class 1A. More than any other antiarrhythmic, class 1A will do it number one. Okay. Next one, clustered, cuboidal, and foamy appearing cells in lungs. What type of cells are these? Foamy appearance. Foamy appearance in the lungs. Under histology, what type of cells are these? Anyone? Nope. Okay. Friction rub after 2 weeks of MI. What is your diagnosis? Anyone? Everyone should answer this very quickly. 2 to 10 weeks of MI. Dressler syndrome. Very good. Okay. Mitochondrial diseases. Which biochemical pathway is inhibited in mitochondrial diseases? Fast answers, please, in the mitochondria. Can I get some answers, please? Mitochondrial diseases, which biochemical pathway is inhibited? Anyone? Which biochemical pathway is inhibited in mitochondrial diseases? Very good. Oops. No, we talked about this before. Okay, we talked about this before, oxidative phosphorylation. Okay. Mitochondrial diseases are two, that is Mellas and Lieber's. Over there, if we do a histological study, we will find there is subsarcolemmal and if we do a muscle study, we will find there's subsarcolemmal and intermyofibrillar accumulation of mitochondria. Subsarcolemmal and intermyofibrillar accumulation of mitochondria and red ragged fibers. Okay. Okay. Okay, last two. Impaired C3 function occurs in which two organisms? Anyone? Impaired C3 function in which two organisms? Anyone? Which two organism impairs C3 function? Nigeria and strepto. Okay. Last question. In HIV with CD4 less than 200, which drug do we give for prophylaxis of toxoplasma and pneumocystis? Anyone? Nope. Which drug do we give? TMP, SMX, trimethoprim, and sulfamethoxazole. Okay. Are we clear about this? TMP and sulfamethoxazole. Okay. Are we clear about the UWorld notes for today? Okay. Okay, so with that being said, can we stop the lecture for here today, because tomorrow is New Year's and today's New Year's Eve, and then tomorrow we can start early since we are going to do classes on New Year's Day, 1st of Jan, 2021. I was thinking of ending the lecture over here right now. Okay, at what time? We will start the lecture at 9 a.m. 9 a.m. Okay. Okay, so with that being said, let me wish you guys a very happy New Year. Hope you guys stay home or go outside. Even if you do, please be safe. Try to stay with your family. Keep me and my family in your prayers. And if there's anything which you guys need, please send us an email and we'll get back to you. And if all goes well, then I'll see you guys next year at 9 a.m. Okay, I'll see you guys next year at 9 a.m. And thank you so much for doing the classes with us. We wish you all the best, and we hope that you guys start the new year with absolute confidence in yourself and in the fact that you are worth so much in your exam. That is the USMLE Step 1. You guys are worth with a very, very good score. You guys are worthy of very, very good scores. Okay. So I wish you guys all the best. If you guys need, please take the day off today. Do not do any questions. Try to stay home and have fun, or go outside and have fun, but stay safe. And thank you for staying with us once again. I will take my leave and I'll see you guys tomorrow at 9 a.m. in the morning. Okay. All right. Happy New Year and bye-bye, guys.